Montalvo in the Biological Sciences Department at the University of Alabama, characterizing the impact of a natural toxin mix in Drosophila species. There are certain species of Drosophila that are able to subsist off of mushrooms. The species that we are particularly interested in are able to subsist off of extremely toxic mushrooms, like the Amanita phalloides mushroom, also known as the death cap mushroom, pictured here on the right. These mushrooms are so toxic that if a human were to eat one, there would be a 100% death rate. There is no known antidote for this type of mushroom poisoning. The mushrooms pictured on the right as well were collected out in the field so that we could use them in our experiments, specifically for mine. The detoxification mechanism of the toxin in these flies is currently not well understood. We do know that in other eukaryotes, the toxin alpha-amanitin, pictured in red on the right, acts by binding to RNA polymerase II, pictured in blue-gray, the large structure on the right. Alpha-amanitin binds to RNA polymerase II, preventing the production of mRNA, which in turn leads to the prevention of production of protein, and this is what leads to cell death in other eukaryotes and eventually death of the entire organism. We do know that these flies do not have a mutation that would prevent the alpha-amanitin from binding to their RNA polymerase II. So somehow the toxin is still binding, but they're somehow able to survive. This is the chemical structure of the toxin alpha-amanitin. It is an extremely large compound, and so it's easy to see why it can wreak such havoc in the body. And amazingly, it is not the only compound that exists in these mushrooms. There are about 9 other toxins that occur simultaneously in these mushrooms, and this is the only one that has been well studied thus far. Since there have been no definite conclusions made in this area, we really want to determine what the mechanism of this tolerance is. And since previous research has only focused on the compound alpha-amanitin, the goal of my project is to determine the effect of the entire suite of natural toxins that occur in these mushrooms on these flies. The first thing we needed to do was determine which species we wanted to use. Of course, we wanted to test tolerant species and susceptible species. And pictured here on the right is a phylogeny of the entire immigrans tripunctata radiation, with the species immigrans on the bottom and the species tripunctata on the top. All of these stars indicate the species that we chose to use. We wanted to make sure that we chose species that were representative of the entire radiation, as well as species that were both tolerant and susceptible. Now that we chose the species, this is our chromatogram of our natural toxin mix. This is a chromatogram of our 10 toxins that exist within these mushrooms. The compounds in red we have known standards for, so we know exactly what they are. The compounds in black we do not have known standards for, but we can be fairly certain that these are the names of the compounds that exist within these mushrooms. We originally started with testing 3 tolerant species and 4 susceptible species, and we collected the larvae and put them into 4 different concentrations of the toxin, one being no toxin, and the other 3 being 5 micrograms, 10 micrograms, and 50 micrograms per milliliter of our amatoxin mix. Once the larvae were put into these food vials, they were checked every day for 30 days, and we checked for their survival to pupation, which is the first life stage after their larval stage. And if they were able to survive as adults and emerge from their pupae, they were collected so that we could measure their adult weight as well as their thorax length. And the thorax length and adult weight we will use to determine if there is a reproductive fitness cost associated with this tolerance mechanism. And we chose 50 micrograms per milliliter for our highest concentration of toxin because previous studies have shown that this is the threshold of tolerance in their studies. Tolerant species should live on this concentration, and susceptible species should certainly die at this concentration. This is the first species that we tested. It's called a melanogaster. It is a susceptible species in a different subgenus called Sophophora. We decided to test this because it has been used as a baseline in other studies. It is well known that it should have very high survival on no toxin and almost 0 survival on any concentration of toxin. And this is shown well in our study here. There was very, very high survival on no toxin and significantly lower concentration on the 5 micrograms per milliliter of the toxin. So we were able to use this as a baseline to continue forward with our experiments. The two tolerant species that we were able to complete were tripunctata, the purple line, and ricins, the teal line. As you can see here, the survival on all of the natural toxin mixes were significantly higher than their survival on no toxin. And you can see by the asterisks above each point in the line, it denotes their significance, with 2 stars being very significant and 3 stars being even more significant. In the end, we tested 6 susceptible species for our study. The first species, Deflecta, the blue line, was the first one we tested, and we were very surprised to see that it survived on natural toxin mix 1 and 2, 5 and 10 micrograms per milliliter, respectively. Our hypothesis was that no susceptible species would be able to survive at these concentrations, and we wanted to make sure that our line had not somehow gained this novel biochemical trait of tolerance, so we decided to test a separate line of Deflecta, Deflecta TW, pictured here. And with this data as well, they survived on the 2 lowest concentrations of the toxin. And as you can see, every single susceptible species that we did test survived on 5 and 10 micrograms per milliliter of toxin. I'd also like to bring your attention to the species Funibris, the purple line at the top. It even had survival on the highest concentration of toxin of 50 micrograms per milliliter. So we were very surprised to see this as well. This is just a numerical representation of the graphs that I just showed. I'd like to bring your attention to the species ricins here. It was one of the tolerant species that we tested. Its survival on no toxin was 0.0267, and its survival on natural toxin mix 1 was 0.267, with a p-value of less than 0.001, showing how significant its survival increase was on the different toxin concentrations. And this is the species Funibris, that susceptible species that was able to survive at the highest concentration of the toxin. As you can see here, its survival on no toxin was 0.72, and its survival on natural toxin mix 1 of 5 micrograms per milliliter was 0.573, which was not a significant decrease. But as you can see on natural toxin mix 3, their survival was 0.107, which was a very significant decrease in their survival. This is the thorax length results that we were able to complete for 2 susceptible species. This essentially shows that there is not a significant difference in thorax length compared either via sex or via toxin treatment, leading us to believe, at least for these 2 species, that there is not a significant reproductive fitness cost associated with living on the toxin. Our next steps for this study include redoing the species multispina, since they, for some reason, did not have any survival on the no toxin food. In addition, we will be completing the thorax measurements for all of the other species to determine if there is a fitness cost associated for any of the other species. Our preliminary conclusions are very exciting. We're showing that there is low-level cyclopeptide tolerance throughout the entire immigrant's tripunctata radiation. Previous studies have thought of this tolerance as either black and white. A tolerant species lives and a susceptible species will die, but we are showing that there is a possible continuum of tolerance that can exist within these different species of flies. And with that, I would like to take any questions.
Would there potentially be a chance, if we understand exactly how these flies survive, to create an antitoxin?
Absolutely. Further along the line, if we were able to determine what exactly the genetic mechanism is, it's possible that we could create a treatment for an antidote.
Other than the creation of an antidote, are there any other future implications of this project?
Absolutely. This is an emerging field of biology. Being able to understand novel biochemical adaptations is pretty new, and being able to understand the different genetic regulation that goes on within each different species would be a real breakthrough in this area of study.
Other questions? Good.